Hi everyone, this is your biology class. I'm your teacher Seema Treja. Student, in this video, first we will discuss the factors affecting the growth and development of the plant. The first factor is temperature. In photosynthesis, we study two categories of plants, C3 plants and C4 plant. Both require different range of temperature for proper growth and development. Whatever the temperature needed by different category of plants, optimum temperature is required. Optimum temperature is required for the efficient working of the enzyme. If the temperature is too low or too high, it inhibits the growth of the plant. The enzymes become denatured or inactive at very low or high temperature. They work efficiently only at the optimum temperature. So optimum temperature is required for the proper growth and development, for the proper working of the enzyme. The next factor is light. Optimum light is needed for the process of photosynthesis as well as photoperiodism. For both the process, optimum light is required. If light is not present, photosynthesis not takes place, photoperiodism, that is flowering, not takes place, so productivity of the plant decreases, no proper growth of the plant takes place in absence of light. Third factor is water. Water is needed for the turgidity of the plant. Cell divide, elongate, vacuolation takes place, protoplasm synthesis takes place. For all this, water is required. Water is required for opening and closing of stomata. Water is required for the photosynthesis purpose. So water play important role for the proper growth and development of the plant. Next factor is oxygen. Oxygen is necessary for aerobic respiration. In aerobic respiration, in presence of oxygen, substrate is oxidized, releases energy, and that energy is utilized for the metabolic activities of the plant. So oxygen play important role in the growth of the plant. Next factor is mineral nutrients. Two types of mineral nutrients are present in nature, macronutrients and micronutrient. Both type of nutrients are essential for the growth and development of the plant. Nutrients help in the synthesis of प्रोटोप्लाज्म की सिंथेसिस के लिए न्यूट्रिएंट्स की रिक्वायरमेंट होती है. तो बोथ टाइप ऑफ न्यूट्रिएंट्स आर essential for the growth and development of the plant. Besides these, one more factor, that is plant growth regulators. Various plant hormones are present: auxin, gibberellin, cytokinin. All these plant growth hormones are essential for proper growth and development of the plant, which we will discuss in detail in our next video. Next topic we are going to discuss here is differentiation. What is differentiation? The growth of plant is indeterminate or open type. Differentiation is an open type of phenomena in case of plants. Like the growth of plant, differentiation is also a open type of phenomena. What is differentiation? Cell divides. ग्रोथ एंड डेवलपमेंट है तो कौन से डिवीजन होंगे? माइटोसिस सेल डिवीजन टेक्स प्लेस. Daughter cells are produced, elongates, these cells elongates. After elongation, they acquire specific shape and size, become specific, mature, and perform specific function. This is called differentiation. Let us discuss differentiation with one more illustration in detail. Here we talk about cell cycle. This is G1 phase, this is S phase, this is G2 phase, and this is M phase. The phase from G1 including S to G2. We called it as interphase. Interphase is preparatory phase in which cell prepare itself for cell division. After completion of the interphase, M phase started. In M phase, cell division takes place. Cell divide and daughter cells are produced. These daughter cells elongates. On elongation, they receive the proper shape and size, becomes mature. This cell escape cell cycle, do not enter in cell cycle further. It escape the cell cycle and enter in G0 phase. The cell present in G0 phase do not divide further. It becomes specialized, becomes mature. For performing specific function. This is the process which we called it as differentiation. Here the newly born cell becomes mature, enter in G0 phase, escape cell size cycle, do not divide further, acquire special structure, becomes mature and perform the specific function. This is the mature cell and the process is known as the differentiation. If this specific cell or mature cell is parenchyma, what special function parenchyma performed in case of plant? Occasionally parenchyma cell store food material. It may become chlorenchyma, participate in photosynthesis. It may become erenchyma in case of aquatic plant and provide buoyancy to the plant organ. Let us talk about dicot stem. In case of dicot stem, what happened? In case of dicot stem, there is present vascular bundle. These are the vascular bundle in case of dicot stem. Vascular bundle in dicot stem is open type. It means cambium is present between xylem and phloem. Above the cambium, there is present phloem, and below the cambium, there is present xylem. The cells present between vascular bundle, compact elongated cell. These are the parenchyma cell. What we call these cells here, medullary rays. They are the special cell. They are the permanent cell, do not divide further. But when secondary growth takes place in dicot stem, at the time of secondary growth, parenchyma cell of dicot stem, parenchyma cell of medullary rays, these are the medullary rays which is parenchymatous. Medullary rays. The parenchyma cells of medullary rays which are in continuation with the cambium cell, they regain the meristematic activity. They regain the power to divide, becomes meristematic, and becomes the part of the cambium ring. At the time of secondary growth, this cambium ring start dividing. On the outer side, it forms secondary phloem cells, and toward the inner side, it forms secondary xylem cell. So what we are observing here, parenchyma cells of medullary rays, they regain the meristematic activity. They acquire the power of division again. This is known as de-differentiation. So at the time of secondary growth, parenchyma cells regain further the power to divide, becomes meristematic, start cutting cells on the outer side secondary phloem in case of dicot stem, and towards the inner side secondary xylem cells are produced. So they regain their lost power of division, becomes meristematic. This process is known as the de-differentiation. After some time, this de-differentiated cell again lose the power to divide and becomes mature to perform specific function. This phenomenon is known as redifferentiation. So there are three phenomena we discuss here. First one is differentiation. What is differentiation? When cell divide and redivide, elongate, differentiate from other, do not divide further, becomes mature, performs specific function. This is known as differentiation. Second phenomena, when this differentiated cell again regain the capacity to divide, becomes meristematic and start cutting cell. This is known as de-differentiation. But after some time, this de-differentiated cell again reach to their original position, again lost the power to divide and becomes specific in function. This is known as re-differentiation. I think the concept of differentiation, de-differentiation and re-differentiation is very much clear to you all. These are the definition given. You can copy from here. What is differentiation? That is, cells divide, mature to perform the specific function. This is known as differentiation. The differentiated cells which have lost their capacity to divide can again regain the capacity of cell division. This is called de-differentiation. After de-differentiation, this cell again differentiated and regain their original position. This is known as re-differentiation. One more topic we are discussing here, that is development. What is development? Development includes all changes that an organism goes through during its life cycle We are discussing here plant. So in case of plant, we are discussing development, that is life cycle from germination of seed to senescence. So all these phases of life started from germination of seeds to senescence comes under the heading of development. Here is a sketch which is given in your NCRT book. Showing the different stages of development, that is cell division takes place, meristematic cell is there, protoplasm synthesis takes place, this is called elongation. After elongation, cell becomes mature and performs specific function, this is known as differentiation. This mature cell undergoes the process of aging, becomes old, this is known as senescence. So all these phases from division of cell to senescence, all we counted under the developmental processes. One more phenomena we are discussing here, that is plasticity. What is plasticity? We discuss plasticity in case of ranunculus, larkspur and buttercup. What we are observing in case of larkspur, two types of leaves are there. In a juvenile phase, small incision are present into the leaf. But when the leaf becomes adult, more incision takes place into the leaf. This is known as heterophily in case of larkspur. That is presence of different types of leaf showing different shape and size. This is known as heterophily. Why this happen? Because plant organs respond to external stimuli or external environmental condition. The plants show different kinds of structure in different stages of life because they respond to environmental condition. This is known as plasticity. Similar in case of buttercup, two types of leaves we can observe. Some of the leaves in case of buttercup are present in the aerial habitat, terrestrial habitat. They are showing different shape of the leaf, and some of the leaf are present inside the water habitat. They becomes more dissected and showing different shape and size. So presence of different types of leaf in different stages of life because the plant respond to different stimuli. This is known as plasticity. Plasticity is also very important from point of view of exam time. I think the concept of plasticity is very much clear to you all. This is all about in this video. Thank you so much.